Hi everyone, this is the first mini lecture of Chapter 13, which is on specific immunity and immunization. In this first lecture, I'll be doing an overview of specific immunity. So specific immunity is the third and final line of defense that we have within our bodies, and it's an adaptive immunity, and it's acquired only after we have been immunized in some way from an infection or perhaps a vaccination. So the two big players in specific immunity are B and T lymphocytes, and these undergo a selective process that gets them ready for reacting to a unique antigen or immunogen. Those are interchangeable, those two words. So what we have with our bodies is what is called immunocompetence, and this is the ability of the body, our body, to react with countless foreign substances. So we have a way to take care of this interaction with foreign substances. So when we talk about specific immunity, we talk about antigens or immunogens, and these are molecules that will stimulate a response by the B and T cells, which are our immune cells that are specific. Antigens or immunogens are, generally speaking, proteins or polysaccharide molecules which are found either on or inside foreign cells and viruses. And any exposed or released protein or polysaccharide is potentially an antigen or something that can stimulate a B or T cell. So foreign molecules that stimulate an immune response this works differently than in the non-specific, where we had PAMPs that were not specific. In this case, these antigens are highly specific and will stimulate a specific immunity. They have to be actually like a lock and key recognition in specific immunity. So the two main features that characterize specific immune response are that specificity, so that ability to recognize a particular foreign agent. In this case, the example would be that antibodies that are produced against the chickenpox virus would not work against the measles virus because it is specific for the chickenpox virus. And the second feature of specific immunity is memory. Lymphocytes have been programmed in our body to remember their first engagement with a particular antigen. And so if we're faced with that antigen again, those lymphocytes will rush to attack, and in larger numbers and quicker. So the response will be more powerful against that antigen. So what does the immune response look like? First, we need to talk about the development of lymphocytes, or the B and T cells. So all of our lymphocytes arise from the same basic stem cell type. One of the big differences is where they mature. B cells will mature in certain bone marrow sites, so a good way to remember this is B cells and bone. T cells mature in the thymus, another good way, that T cells and thymus, so both start with a T. And both of these cell types, both B cells and T cells, will migrate to lymphoid organs, although they will be in different areas within those lymphoid organs. In addition, B and T cells constantly are circulating throughout our circulatory system and our lymphatic systems, and they migrate in and out of our lymphoid organs. And this is their way of surveilling or looking for foreign agents. In your book, there is a nice table with an overview of the stages of lymphocyte development and function. So starting on the left-hand side, we have the B cell line, and on the left side, you have your T cell line, all starting from the same origin, but following a different pathway. This is a good one that reiterates the things that I spoke about in the earlier slides. And then what happens, though, with these mature cells when we have an antigen enter and present itself? So when a pathogen comes in, it's carrying antigens that have made it across that first line of defense, right? It has proteins or polysaccharides that have made it through that first line of defense. And what will happen is our body will recognize that, and it will send macrophages to that site to try to attack this particular agent. And these macrophages will ingest the agent, the foreign agent or pathogen, and start the inflammatory response. When dendritic cells ingest the pathogen, they will process that pathogen and will actually take portions of it to present on the outside of their cell to T lymphocytes for them to come along and look at it, and that will start this cell-mediated immunity. In addition, there will be pieces of that pathogen which will drain to the lymph nodes, and these antigens in the lymph nodes can activate B cells. So how is it that our T cells can recognize these agents when they've been digested by phagocytosis or macrophages? All cells have a variety of different markers on their surfaces, and this is true for our cells as well. And this allows for detection. So if it's a foreign agent, it would allow us to detect that. It allows for recognition of perhaps cells within our body that would be recognized as self or ones that are not foreign. And then cell communication, and we saw this earlier with our different ways that cells can communicate. These markers will play an important role in activating the different components of the immune system. So the main one that we talk about when we're looking at specific immunity is the major histocompatibility complex, or MHC. And this is a set of genes that codes for human cell markers or receptors. So they're found on all of our cells except for red blood cells. They can also be known as human leukocyte antigen system, or HLA, but in this case we really talk about them as MHC. And these markers play a vital role, so they allow us to recognize cells that are self, or ones that are not foreign to us. And then they also allow us to recognize things that are foreign. So in the case of a foreign tissue, allow us to do rejection because they're not recognized as self cells. So the major histocompatibility complex can be divided into three classes. The class 1 genes will code for markers that appear on all nucleated cells, and they display that characteristic of self and allow recognition of the immune reactions. We also have class 2 genes which code for immune regulatory markers found on different types of immune cells, macrophages, dendritic cells, and B cells. And these are the ones that are involved in the presentation or the taking of those pieces of foreign cell to present to T cells. And then last, we have class 3 genes, which encode proteins involved with the complement system. You'll remember the complement system was one of our non-specific antimicrobial protein systems we talked about in the last chapter. So these MHCs will allow those antigens to be presented, and they will be presented to lymphocyte receptors. So both B cells and T cells have receptors on them, that their major role is to accept or grasp or bind those antigens in some form. So you have this foreign agent that's come in, it's been digested by a macrophage, and it's been presented. It also may have broken down those pieces, and some of them flowed into lymphoid tissue. In the case of the ones that have flowed into lymphoid tissues, B cells have receptors that will bind to those free-floating antigens. In the case of T cells, they actually need to have them presented by the MHC molecules on the cell that has digested them. So T cells have receptors that will bind to the antigens after they've been processed and presented on the cell surface through the MHC molecule. And the receptors of these lymphocytes have the capacity to respond to nearly infinite numbers of unique antigens. So any number of antigens, we should have a specific receptor on one of our lymphocytes. And I'll talk about what happens after that receptor recognizes and binds to a unique antigen, what then follows in the immune system. And that'll be part of our next lecture on this chapter.